Hello students, let us start online class in botany. At first, about our syllabus. In our biology textbook, there are 16 chapters. Of these 16 chapters, 8 chapters are of botany syllabus. So these are the name of these 8 chapters. First chapter, reproduction in organisms. Second chapter, sexual reproduction in flowering plants. Third chapter, strategies for enhancement in food production. In that chapter, we have to learn about plant breeding. That means one part of agriculture, and also about animal breeding. Then fourth and fifth chapters are of biotechnology unit. Remaining 3 chapters are of ecology units. So you have to learn 8 chapters in botany syllabus, and for easy handling of online class, I start with the second chapter. Name of second chapter is sexual reproduction in flowering plants. I start with this chapter because you learn so many important points in the sexual reproduction of flowering plants in your first year syllabus. Sexual reproduction is the reproduction by the fusion, by the union of gametes. And now we have to learn sexual reproduction only in flowering plants. So we have to learn reproduction by the fusion of gametes in flowering plants. For a flowering plant, there are 4 main organs. They are root, stem, leaf, and flower. Flower is the organ for sexual reproduction. That means gametes are produced by flowers. So we have to learn the structure of flower. This is the diagram showing cross section of a flower. In flower, different parts are arranged as whorls. Whorls means circle. Different parts are arranged as whorls. There are mainly 4 whorls in a flower. The outermost whorl is calyx. Calyx, it is the outermost whorl. In this diagram, sepal is given. It is one member in calyx, sepal. Then second whorl is corolla. Corolla, it is the second whorl. In the diagram, it is shown in the form of petal. Petal means member in corolla. Third whorl is androecium. It is formed of many stamen. Androecium formed of many stamen. And the innermost whorl is gynoecium. Gynoecium is formed of many members known as carpels. Regarding sexual reproduction, there must be formation of spores. There are 2 types of spores. They are smaller spores and bigger spores. Smaller spores are known as microspores and bigger megaspores. Smaller spores, microspores, are produced by androecium, and megaspores are produced by gynoecium. So let's learn androecium at first. So you can observe 2 diagrams. First one, it is the member of androecium. Its name is stamen. Member of androecium is stamen. Stamen has mainly two parts. There is a long stalk for stamen. Long stalk. Its name is filament. The stalk of stamen, filament. And at the terminal portion, there is a bilobed structure for stamen. Bilobed means two-lobed, two-part. So at the terminal portion of stamen, we can observe a swollen two-lobed part. That part is anther. And there are two lobes for anther. Within each lobe, there will be two cavities. Cavities are known as theca, and these cavities in anther lobe will produce microspore. The cavity producing microspore is microsporangium. Microsporangium. Angium means vessel or cavity. So the cavity producing microspore is microsporangium. When this microsporangium becomes mature, another name is given to it. It is pollen sac. Pollen sac. And within an anther, there are two lobes. Within each lobe, there will be two microsporangia. So within one anther, there will be total 4 microsporangia. Within microsporangium, there will be production of microspores. So we have to learn the structure of microsporangium. So in the second diagram, at the cut plane, we can observe 4 microsporangia. To learn the details, observe these figures. In the first figure, 4 wall layers of microsporangium are labeled. 4 layers in the wall of microsporangium. The outermost layer is epidermis. Epidermis. Second layer, endothecium. Third type of layer, middle layers. There will be 2 to 3 layers of cells in middle layers. Then the innermost wall layer of microsporangium, it is tapetum. Look at the diagram at the right side. We can observe cells in these 4 wall layers. So refer 4 wall layers in that diagram at the right side, diagram B. Epidermis, endothecium, middle layers. In the middle layers, we can observe 2 layers of cells in this diagram and tapetum. Now we have to learn functions of each of these layers. Actually, for the first 3 types of layers, there is same function, and they have 2 common functions. One, protection of microsporangium. Second one, dehiscence of microsporangium. Dehiscence means breaking. Why the microsporangium breaks? It breaks open when it becomes mature. It is to release microspores. Because microspores only after their release will reach near female gamete. So there must be dehiscence of anther to release microspores. So the 2 functions of these 3 layers, epidermis, endothecium and middle layers, 2 functions. One, protection of microsporangium, and the one, dehiscence of pollen grains, that means microspores. Dehiscence of anther to release pollen grains, to release microspores. Then the innermost layer of the wall of microsporangium, it is tapetum. The main function of tapetum, it is different from the function of other layers. That function is giving nutrients, giving nourishment, giving food molecules to the cells of microsporangium. So the function of tapetum is the nourishment of microsporangium. And within microsporangium, we can observe a young tissue. Name of that young tissue is sporogenous tissue. Sporogenous tissue. And the cells in this tissue, after some time, undergo division to form microspores. The cells of sporogenous tissue are diploid cells. They undergo division to form haploid microspores. So if diploid cells produce haploid cells, you know the type of cell division required, it is meiosis. So the cells of sporogenous tissue undergo meiosis and haploid microspores are produced. This process is called microsporogenesis. Microsporogenesis. It is the process of formation of microspores. And we have to give a name for these young cells undergoing meiosis to produce microspores. Actually, these cells are the parents of microspores. Actually, the mother of microspore. So these cells are known as microspore mother cell. Microspore mother cell. And the name pollen mother cell. The mature microspore is known as pollen grain. So the microspore mother cell is also known as pollen mother cell. Short form PMC. PMC. So this pollen mother cell undergoes meiosis to form microspores. This process is microsporogenesis. And if one cell undergoes meiosis, products are four cells, four daughter cells. So when microspore mother cell undergoes meiosis, four microspores are formed. They are known as microspore tetrad. Microspore tetrad. And later these four microspores will separate from each other, and when they become mature they are known as pollen grains. Pollen grains. There is another name for pollen grain, that is male gametophyte. Male gametophyte. What's the meaning of phyte? P-H-Y-T-E. Phyte. Phyte means plant. So the plant producing microspore is named as microsporophyte. So the pollen grain, according to scientists, is a plant. Each pollen grain is a plant. And that plant produces male gamete. So pollen grain is also called male gametophyte. So let's learn about this pollen grain. So when you observe pollen grain under microscope, there will be different appearance for them based on the species of plant. And if you are an expert in observing the morphology of pollen grain, you can identify the species of the plant from which pollen grain is formed, because for each species of plant there is a specific appearance. There is a special appearance for their pollen grain. And regarding study of pollen grain, at first take the cell wall of pollen grain. Cell wall of pollen grain has two layers, one outer layer and one inner layer. Outer layer of cell wall of pollen grain is thick and it is known as exine. It is exine. And inner wall of pollen grain is thin, it is intine. It is intine. So exine and intine are the two layers of cell wall of microspore. Exine is formed of a chemical known as sporopollenin. Sporopollenin. Peculiarity of this chemical, it can withstand high temperature and also low temperature. Usually it does not react with highly concentrated acid or base. It is not digested by enzyme. That means this sporopollenin is long-lasting molecule. It is one of the most resistant biochemicals. That's the peculiarity of sporopollenin. It is one of the most resistant biochemicals. So this pollen grain covered with this resistant chemical, it can remain at different part of earth for many years, for many centuries. So the pollen grain of many plants, many ancient plants are naturally preserved as fossil. So the pollen grain of many ancient plants are naturally preserved as fossils due to the presence of sporopollenin as a cover on this pollen grain. Then on this exine, we can observe some openings, some apertures. Aperture means opening. These apertures on exine, on this pollen grain, are known as germ pores. Germ, germ pores. Actually, full form germination pore. During maturation of pollen grain for the release of male gametes, one tube formed out of come out of pollen grain through this germ pore. That is why these pores are named as germination pores, germ pores. And the inner layer of cell wall of pollen grain, inner layer, its name is intine. It is intine. It is formed of the common chemical in the cell wall of plant, cellulose and pectin. So cellulose and pectin are the two main chemicals in intine. Then about the development of pollen grain from microspore. So microspore is haploid. It is formed by meiosis of pollen mother cell. So at the time of formation, there will be 4 microspores formed together, and they are kept together for a particular period of time. At that time, they form a tetrad of microspores that is given in this figure. Tetrad. Tetrad means 4 cells. Tetra means 4. So observe this tetrad of microspore. Then these 4 microspores separate from each other. This is a single microspore. Then this microspore further develops. Then it undergoes division. It is mitosis. As a result, 2 cells are formed. 2 cells. One is big, other one is small. Big cell is vegetative cell. Vegetative cell. And the small cell is generative cell. Generative cell. So the anther breaks and microspore. Now this mature microspore, pollen grain, that is released at this stage. When the pollen grain is in 2-celled stage, it is released in many plants. Then refer the generative cell. This small cell undergoes further mitosis. It undergoes one more mitosis and 2 cells are formed. These 2 cells are the male gametes. So after the formation of male gametes, what's the number of cells in this pollen grain? 1 vegetative cell and 2 male gametes. So total 3 cells. In some plants, pollen grains are released when they are in this three-celled stage, only in some plants. So this is about the structure of pollen grain. At first about the wall of pollen grain with exine and intine, then about the inner part of pollen grain, at first with a single cell, then with vegetative and generative cells, then the third stage with one vegetative cell and two male gametes. Remember, and the name of pollen grain, it is male gametophyte because it contains two male gametes. So this is the first part of presentation. Then there is one assignment for you. It's a question. You have to answer this question in your notebook. Our question: which are the four wall layers of microsporangium? Which are the four wall layers of microsporangium? Write their functions. Okay, now we can conclude this first session of online class in botany. Thank you.